I only have one conflict, which is not really a conflict other than an academic conflict. I'm an employee of the US government, so we're scrubbed pretty cleanly. I've been the director of NICHD for a little less than 2 years, and I made this slide because people kept telling me, you're different. And other than the obvious, somebody else told me that I was the first NICHD director with a full head of hair. So I made this slide because I didn't know if it was true, and it is true. But the other thing is I'm the first director in the last 30 years to actually have come from a research background. So I was a professor at Tufts University School of Medicine, had my own laboratory there. And so while being director of NICHD is more than a full-time job, I still have a research laboratory which is in the Human Genome Research Institute at NIH. So it's my great pleasure to walk across campus where I can meet with my lab team and still continue to move the field forward in some small ways. So our laboratory focuses on prenatal genomics and on fetal therapy, and my hope is today that you'll learn something that perhaps you didn't know about. Well, we all know that society is changing and there are many disruptive technologies. So we no longer hail a taxi, maybe here we hail a taxi, but in many parts of the world we hail an Uber. The younger generation can barely read maps anymore, they just look at their iPhone. Many people stay in Airbnbs rather than hotels, but I'm going to talk about a disruptive technology in prenatal testing, and that is sequencing of cell-free DNA that circulates in the blood of pregnant women. Well, prenatal genomics is probably the #1 implementation of genomic medicine. Eric Green, who's my colleague, who's the director of the, the National Institute, Human Genomics Research Institute, when he was asked about the successes of the Human Genome Project on its 15th anniversary, said that it was prenatal genomics. I'm going to talk to you about prenatal screening for Turner syndrome prior to non-invasive prenatal testing, or NIPT. There are multiple different terms that are used: non-invasive prenatal testing, non-invasive prenatal screening, cell-free DNA sequencing. I'm going to talk about prenatal screening in the NIPT era, and then I'm going to talk about mechanisms for underlying false positive and negative NIPT results for Turner syndrome. And then talk a little bit about the possibilities of fetal therapy, because as I'll show you, some of the pathology is actually very visibly present in utero. Well, we don't normally screen for sex chromosome aneuploidies with the biochemical testing that has been part of the standard of care for the last 25 years. Those tests are mainly directed at autosomal aneuploidies, not sex chromosome aneuploidies. Although they are considered to be the standard around the world, I'll show you that their positive predictive values are not very good. So the way that we find out about Turner syndrome prenatally is through prenatal sonography. And there are a number of findings that are virtually diagnostic of Turner syndrome, and these include an increased nuchal translucency, but specifically the cystic hygroma. Also, this is very high in the differential diagnosis of fetal hydrops, as well as renal and cardiac malformations. But as a geneticist, I've taken care of many young girls with Turner syndrome who were not recognized in utero. So what's happening now, and it's really been a revolution that has occurred over the last 20 years. In 1997, it was first recognized that cell-free DNA was circulating in the blood of pregnant women. Now, this is DNA that is coming from the placenta, and it's highly fragmented. It is mixed with the DNA from the mother. So if you take a blood sample, you're getting this cell-free DNA from apoptotic cells. What we're going to call fetal DNA from here on in is actually placental DNA. And when we do the sequencing, it's performed on a mixed DNA sample. So the average sample, if it's taken late in the first trimester of pregnancy or early in the second trimester of pregnancy, it contains around 90% maternal cell-free DNA and around 10% placental or fetal DNA. So you are sequencing maternal and fetal DNA at the same time, which is going to become important later on. You basically cannot distinguish between the maternal and the placental DNA unless the baby is a boy, and there will be Y chromosomal material there, or if there are specific single nucleotide polymorphisms or specific gene mutations that the fetus has that the mother doesn't have, and also the fragment sizes are a little bit different. Where do these cells come from, these apoptotic cells? Well, if you start with the undifferentiated cell, let's see, I'm trying to get the arrow in here. Here we go. We are looking basically at the DNA of the chorionic ectoderm, which is over here, mixed in with the maternal cells. The fetus is over here, so this material is actually quite distinct embryologically from the fetus. The chorionic mesoderm, which is what's sampled with a chorionic villus biopsy, is very similar to what is in the cell-free DNA sample. Now, what really changed the game occurred in 2008. So you know that sequencing has progressed. Sequencing techniques have progressed over the last 30 years. And what happened in around 2007 was that massively parallel sequencing became commercially available, and it was affordable so that individual laboratories could buy massively parallel sequencing machines. And Stephen Quake from Stanford, who is on the left, and Dennis Low from the Chinese University of Hong Kong on the right, within a few months of each other, published studies that suggested that massively parallel sequencing techniques could be used as a counting system instead of looking for a single base change. And that really changed the game. So, excuse me, as an example, you have this fragmented placental and hematopoietic DNA in the maternal plasma. As I told you, they are mixed. So this is representing the baby's DNA, this is representing the mother's DNA. These fragments are about 150 to 200 base pairs long. You only need to sequence the first 25 to 36 base pairs of the fragment, and that is unique enough to map it to a particular chromosome. And the way this testing works is you count the number of fragments that are mapped to a chromosome of interest, for example, chromosome 21, and you compare it in a ratio to another chromosome or a set of chromosomes. The ratio should be 1:1. If there is too much 21 relative to the chromosome of reference, it suggests that the fetus has trisomy 21. If there is too little X chromosome compared to the reference, which is usually a female, it would suggest that the fetus is either a male, so you would look for the presence of a Y, or the fetus has monosomy X. Well, this has been employed to really change the game in prenatal screening for trisomy 21. And this is from a meta-analysis done by a group in the UK who looked at the three most common autosomal aneuploidies: Down syndrome, Edwards syndrome, trisomy 18, and Patau syndrome, trisomy 13. These are in high-risk women now, looking at 22 studies, 19 studies, and 11 studies, respectively. And you can see that the meta-analysis showed that the cell-free DNA sequencing screen has a positive predictive value of 91% for Down syndrome. So it is nearly diagnostic, but it is a screen. There is nothing that performs quite like this. It's so good that people said, well, why don't we use it as a primary screen? So we and others have directly compared in a general obstetric low-risk population, how does DNA sequencing perform to the current standard of care, which is biochemical testing for proteins produced by the fetus and placenta and an ultrasound measurement of the nuchal translucency. The current standard of care in both studies was around 3 to 4%, the positive predictive value, which means that we get a lot of false positives. If you have a positive screen with the current standard of care, it's only 4% chance that the fetus really does have trisomy 21. There's a 96% chance that it's a false positive. Even in a low-risk population, our study showed that there was a 62.5% positive predictive value, and in another study, they showed an 80.9% positive predictive value as a screen. But what has really made the difference is the negative predictive value is greater than 99%. So what this means is that women who are high risk for a fetal autosomal aneuploidy, for example, women over 35, if they find out that they have a negative NIPT test, they don't want to have an amniocentesis. So the current recommendation for obstetrics have changed significantly since this testing became available. The testing only became available in 2011, and there's really been this enormous change in practice, and this really shows the clinical utility of the testing. So NIPT is offered to women as an alternative for an invasive procedure for women who are high risk, women over age 35, women with a positive family history of Down syndrome, women who have a fetus with sonographic abnormalities suggestive of an autosomal aneuploidy. But if the screen is positive, it's universally recommended that you have a diagnostic test to confirm. Low-risk women around the world, and I did check with a few people here in Greece, they are having the test, but generally they are self-paying for it. In the United States, it's very common for middle-class and upper-class women to have the test, even though their insurance won't pay for it. And then they have gender reveal parties. So they open the envelope with the NIPT results, and then they all celebrate if it's a boy or a girl. We recommend that pre- and post-test genetic counseling be provided for all so that women understand that these results will say something about their own DNA as well. There's microdeletion testing that costs more, and then there's the sex chromosome testing, which honestly is why most people have the test. They want to know, are they having a boy or a girl very early on in the pregnancy. There's no extra charge, but one result is that you can determine whether or not the fetus has a sex chromosome aneuploidy. So what does that look like? Well, here's a series of cases where you can see that this would be the reference line here, and this is the normalized chromosome value, which is akin to the Z-score, and fetuses with monosomy X have a lower than reference. And it's shown in a different way here. The monosomy X zone has low amounts of X with no discernible Y. Well, the difference with testing for Turner syndrome is that the positive predictive values are much lower than they are for Down syndrome. I'll tell you a little bit about our study in a moment, but look at the positive predictive values in this column. They only range from 20 to 40%. I think they're probably closer to 20-25%. And because it has been shown that you can have chorionic mosaicism for 45X, the most appropriate follow-up test is an amniocentesis. Well, there are a variety of mechanisms for these underlying results suggesting monosomy X. The thing that's unusual about monosomy X is it's not associated with maternal age. It occurs mainly because of post-fertilization non-disjunction. So these fetuses start as 46XX. You can have false positive results where the mother is actually a Turner syndrome mosaic, or you can have maternal somatic mosaicism, which I'll discuss in a moment. You can have confined placental mosaicism, where part of the placenta has 46XX and part of it has 45X. You can have a twin that died in utero, and you would still have material from the placenta circulating in the maternal circulation. And you can also have false negative results. We did a study in collaboration with a large clinical laboratory of 18,161 tests, and the test results were concordant if the result matched what was known about the fetal or neonatal karyotype or birth outcome, or discordant if they didn't match. No discordance was reported if we had no follow-up information. For sex chromosomes, the XX and XY, this was a different part of the study, discordant cases were divided into the ultrasound and the karyotype groups. What I want you to see is in this cohort of 18,000 plus samples, there were 204 cases of sex chromosome aneuploidies detected non-invasively. The majority of them involved the X chromosome. Detection of the Y chromosome is generally very precise, and again, it's not affected by the fact that the mother, in most cases, doesn't have a Y chromosome. There are some cases where she can. Most of the discordant cases involved monosomy X. There were also cases of triple X. What's the mechanism for the false negative cases? Well, as you know, most cases of Turner syndrome do not survive gestation. It's very common to have conceptions that in the early stage are 45X. And as I said earlier, this is not associated with advanced maternal age. But Hook and Warburton, who are cytogeneticists, concluded that a very large proportion of 45X embryos acquire their 45X after fertilization. And they hypothesized that the only reason why you get some embryos and fetuses surviving gestation is that they are mosaic in the placenta, and the placenta is supplying a critical gene known as PSF2RA, which is critical for placental function. So the placenta in these cases is serving as a rescue for the fetus. Now, what's fascinating, what our laboratory has spent a lot of time on, are the maternal incidental findings, which generally come as a surprise because in most situations, the pre-test counseling does not talk about the possibility of the mother finding out she has something. In China, there was another study that showed they tested 47 triple X women who had 47 triple X results or 45X results, and then got the mother's Buffy coat, which we don't normally get. We're just analyzing the placenta. In almost 9% of cases of these sex chromosome aneuploidies that were revealed by the NIPT, almost 9% of the cases were due to a maternal X chromosome abnormality. So they have recommended mandatory maternal white blood cell testing after the finding of a potential sex chromosome abnormality. This is not trivial. I recently spoke at a Turner Syndrome Research Society meeting, and they were telling stories about how some obstetricians are counseling women to terminate after a prenatal testing result of Turner syndrome. Now, with the very low positive predictive values, I would very much argue against that. And the first step you should do is take a history, which I'll show you in a minute. And the second step is you should take the mother's blood. But the other thing that's really fascinating is we've learned that many more fertile women are out there with Turner syndrome mosaicism than previously recognized. Now, we don't know if all of them are actual constitutional mosaics or whether they are constitutional mosaics or whether they are somatic mosaics. There have been several studies looking at the percent of cells with a single X chromosome in women as they age. And there is no doubt that in white blood cells you can lose one X chromosome the older you get. And remember that many of these women are over age 35, maybe they had difficulty conceiving, they don't want a diagnostic procedure because they don't want to risk a miscarriage. So it's very common to see somatic mosaicism in this older population of women. And in fact, in our study, we showed there was an association between monosomy X and maternal age, but we did not see that association in the women who had 47 triple X. So some of the things you can do right away if you get this kind of result is ask the woman, are you over age 35? And in that case, she's a candidate for somatic loss of the X chromosome due to aging. Believe it or not, I find this really hard to believe, but we know of women who've had kidney transplants or bone marrow transplants where their obstetrician doesn't know that history. So you get an NIPT result of 46XY. The fetus looks female on ultrasound, and everybody freaks out, and then you find out that the woman had a kidney transplant, and what you're actually detecting is DNA from the kidney that is being released into the maternal circulation. So the kidney donor was a male, there's XY material in that kidney. You can ask if the woman's height is significantly different from her parents or siblings. And then you can ask, did you conceive via assisted reproductive technology? And again, this material is not necessarily in the history because it may be a reproductive endocrinologist with one set of charts and maternal fetal medicine specialist with another set of charts. But we do know of at least one woman who had 46XY, again with a female fetus, and she conceived with egg donation, and she had Swyer syndrome. So she was 46XY herself. So again, neglected to tell anybody, we would have expected that her DNA results show 46XY. Now there's a big question, now that we know that there's so many of these women out there, we have no idea how they should be managed. And this is something that we would like to study, hopefully in the near future. How best to distinguish between somatic and constitutional mosaicism. Importantly, since these women are pregnant, and women who have Turner syndrome who are pregnant are at increased risk for hypertension and preeclampsia, and importantly, aortic complications, should we worry about the women who are newly diagnosed as mosaics, and what kind of workup should they have? Should these women deliver in a tertiary medical center? If this is just somatic mosaicism due to aging, probably not. But if they are truly constitutional mosaics, then yes, absolutely. And then postpartum, should they be treated as an adult woman with Turner syndrome? Should they be monitored for lipid, thyroid, and bone mineral density abnormalities, or should they not? We do not know the answers to these questions. So I just, in the closing minutes, wanted to suggest some possible future approaches to antenatal therapy of Turner syndrome using transcriptomics. And my laboratory for a long time has been studying cell-free RNA now, not DNA, but RNA in the amniotic fluid. And we started studying the cell-free RNA because it was a relatively pure sample. We knew that in the maternal plasma, it's a mixture of maternal RNA and placental RNA. So, and we also knew that before there was this global decrease in the number of amniocenteses, there was a lot of amniotic fluid around with known karyotypes. So we began getting the discarded amniotic fluid and studying cases versus controls. And we found that the RNA in the amniotic fluid provided a real-time window into fetal development, changing daily and weekly, but also there were transcripts from multiple organs in the amniotic fluid, including the brain, the thyroid, this jumps around, the lung, the liver, etc. So it really gave you a complete picture of what's going on with the fetus. So we've done a number of studies, and we have the dysregulated gene lists that are all available and all publicly shared via the Gene Expression Omnibus. If anybody wants to look at these data, they're easily available. And what we found when we compared fetuses with 45X to fetuses with 46XX that were matched for gestational age, there were 470 statistically significantly differentially expressed genes. Not surprisingly, EXIST was significantly downregulated. But what was fascinating was there were certain genes that were quite significantly upregulated that are involved in areas of pathology that you see in children or women with Turner syndrome. So we saw an upregulation of NFAT-C3, which is involved in perivascular tissue remodeling. We saw an increase in the LDL receptor, which is associated with hyperlipidemia, and an increase in IGF-FBP5, which reduces growth and fertility. And SHOX has been postulated as an important gene in Turner syndrome, but we found absolutely no differences between cases and controls. So interestingly, this points to some potential targets for therapy prenatally. Also, we looked at where these particular transcripts mapped, and we saw an overrepresentation of immune dysregulating genes. So it made us wonder whether some of the antenatal pathology seen in living fetuses with Turner syndrome was actually related to autoimmune processes. And because we've been studying different aneuploidies over time, we can say that each aneuploidy has a completely unique pattern of gene and pathway dysregulation. So the focus of the laboratory has really right now been on trisomy 21, where our therapy targets are more related to oxidative stress and neuroinflammation, and we're hypothesizing that if we treat antenatally, we can improve neurocognition. But Turner syndrome is going to be our next target because what's fascinating is you see these abnormalities in cholesterol homeostasis, evidence of autoimmune disease, and what we think is the predecessor to the coarctation of the aorta. You're seeing this at 15 to 16 weeks of gestation. Very clear, you don't need to be a geneticist or a bioinformatician to see the difference between a Turner syndrome fetus and a fetus with normal chromosomes. So you can take those dysregulated gene lists and plug them into a database called the connectivity map, which is a reference database of about 1,500 FDA-approved molecules that could be potentially used to treat some of these pathways in utero. We haven't done it for Turner syndrome yet, but we have done it for Down syndrome. And you can test hypotheses to say, using this database, can you essentially reverse the pathophysiology of this condition? So I don't have time to go into all of it, but what we're looking for are very, very negative scores. We're looking to reverse the pathophysiology of Down syndrome. And so in one study, we looked at a variety of different FDA-approved drugs and looked in 9 different tissues from fetuses with Down syndrome, as well as some mouse models that we're studying that have the equivalent of Down syndrome. And we're looking for molecules that are highly negative. The connectivity map goes from +1 to -1, 0 is neutral. So because apigenin, which is a naturally occurring flavone, has the most negative score across the highest number of tissues, we are just completing now a prenatal therapy trial of mice, the TS1 CJE mouse model of Down syndrome, and we have shown some therapeutic effects, both on development in terms of specific behaviors, as well as in human amniocytes, showing that this reduced oxidative stress. So we're very enthusiastic about our hypothesis testing, and we believe that we could do the same thing antenatally for Turner syndrome and perhaps retain fertility or perhaps try to minimize the amount of cardiovascular disease. So in summary, cell-free DNA sequencing has already had a profound effect on prenatal clinical care. It's led to about a 70% global reduction of invasive diagnostic procedures. There are a lot of maternal fetal medicine specialists who are very angry with us because they don't have the amount of procedures that they used to have. But on the other hand, women are having safer prenatal testing. The positive predictive values are very high for trisomy 21, but are much lower for Turner syndrome. As I've told you, the sequencing analyzes both the maternal and the placental DNA. The false positive results generally have underlying biological mechanisms. For example, we can also detect cancer. When the mother has cancer, we pick that up with this testing. But we detect pregnant women with mosaic Turner syndrome, and this provides an unbiased opportunity for all of you to study the natural history of women with Turner syndrome. And if you're interested in collaborating on that, we will be in a year or so, hopefully, starting a study. The follow-up management of these mothers with 45X variants needs further study. And by accumulating a registry, hopefully we'll be able to provide some information. And lastly, for me, the future has always been about treatment, not about diagnosis. And analysis of the fetal transcriptome suggests that there is an antenatal onset of many of the pathologies associated with Turner syndrome. So there's the potential for future therapies that could be given to the mother that crossed the placenta to preserve the ovaries and prevent cardiovascular disease using dysregulated gene expression and pathway analyses. And I just want to leave you with this beautiful sculpture from Groningen in the Netherlands of the X chromosome and the Y chromosome. And it looks like the Y chromosome is bowing to the X chromosome. Thank you very much.